Securing food for the next generation undoubtedly comes with paramount challenges. In today's world, it is critical we use ethical and sustainable practices to produce high-quality protein and nutrients for the global population. Scientific innovation, especially when pertaining to genetics, presents opportunities to optimize animal production practices, which promote animal, human, and environmental health. Today, we will demonstrate how the process of embryo transfer and advanced embryo health analytics can contribute to global food production goals. Embryo transfer is the process of removing one or more embryos from the reproductive tract of a donor female and transferring them to one or more surrogates or recipients. Donors are DNA tested, and only those with the genetics to improve animal health, efficiency, and sustainability are selected as donors. Ideal donors possess a normal reproductive tract, are free of conformational defects and genetic abnormalities, and express desirable heritable traits such as increased milk production, feed efficiency, carcass and meat quality, or insect resistance. We want to collect as many embryos as possible from these donors so we can perpetuate her genetics to many offspring each year. In preparation for the embryo transfer, the estrous cycle of the donors and recipients are synchronized with exogenous hormones, so the recipient will be receptive to becoming pregnant when the embryos are transferred. In the weeks leading up to a conventional transfer, the donor received combinations of hormone injections and induced a superovulatory response to encourage multiple eggs to ovulate, so we have more opportunities to create more of her genetically superior offspring. Estrus is induced in the donor, and then she is artificially inseminated with high genetic value frozen semen, and we allow 7 days for the eggs to become fertilized and travel from the oviduct to the uterus. Now we will perform the embryo recovery, or flush. Dr. Killingsworth loads the donor into the squeeze chute so she can be safely restrained for the non-surgical procedure. She is given an epidural to relax her pelvic region, so we can access her uterus transvaginally to flush and retrieve all available embryos. The reproductive anatomy of a cow consists of a bicornuate uterus, which means the uterus is divided into 2 prominent horns, which are each associated with an ovary. It is important each horn is flushed to ensure all embryos are recovered. Dr. Killingsworth can access each horn by inserting one hand into the rectum and then feeling through the rectal wall to identify the reproductive structures below. He can physically hold the cervix to guide the catheter through the vagina and cervix to properly seat the catheter in one horn of the uterus. In cattle, this can be tricky because the cervix has annular rings of cartilage, which makes passing a catheter through the cervix into the uterus feel like a labyrinth. However, he can verify the successful insertion by feeling it with his other hand. Once the catheter is in the proper position, saline solution is allowed to flow into the uterus. A Y-shaped connector is used to allow the media to flow into the uterus and then back out another line and into a collection cup with a filter to trap and recover embryos. During this process, the veterinarian will massage the uterine horn to encourage the release of all embryos with the return of the fluid. Once the veterinarian is satisfied that the horn was properly flushed, he will reposition the catheter into the other horn and repeat this procedure on the other side. We will not know if Dr. Killingsworth successfully collected any embryos until we get into the laboratory, because they are too small to be located with the human eye without a microscope. At this point, the donor cow is free to be released, and we will carry the embryo cup into the laboratory, empty the contents into the search dish, and search for embryos under a microscope. The veterinarian or embryologist will diligently search the dish looking for embryos. Oftentimes, there are traces of blood or mucus in the dish, which make it difficult to find the embryos. When an embryo is found, it is picked up from the dish with a pipette and moved into a new, smaller dish with clean holding media. Once all embryos are located, they are examined at a higher magnification and classified according to their development stage and morphology. This is a subjective analysis, which is solely based on the examiner's ability to evaluate visually apparent characteristics of the embryo. This method can effectively identify embryos which are infertile, have stalled in development, have significant cellular fragmentation, or extruded cells, but it cannot relay information pertaining to morphokinetics, which are known important factors relating to an embryo's health and viability. To gather further information regarding the embryo's morphokinetic activity, such as development rate, metabolism, and stress, we will use the EmGenisys evaluation to scan each embryo for signs of life. While the embryos are still in their holding dish, we arrange them in a line in which the embryos are not touching or superimposed. We then use our smartphone to record a 30-second video of the embryos and upload the embryos into our web-based platform. This analysis can be performed immediately to scan each video frame by frame, measuring the embryo's real-time morphokinetic activity to provide a comprehensive evaluation of the embryo's health. Then, the veterinarian can access the reports and use this objective data to manage his breeding strategy. When the healthy embryos are identified, they can be frozen for later use or transferred immediately as fresh embryos. With this technology, we will unlock the potential of next-generation animal production. Join us. We are breeding something epic.